Hey guys, welcome back to another video. So in today's video, we're going to be talking about blood glucose concentration, metabolism, and also how to control how the body controls blood glucose concentration. Now, this entire thing is based on negative feedback, where the cycle goes as if there's if there's a stimulus, the receptors and sensors will sense the change in whatever's happening. It will send afferent signals back to the central nervous systems, and from then on, the central nervous systems will send efferent signals to the effectors, or which are the muscles or the glands, and then a response will be carried out. So in this case, if blood glucose concentration is too high or low, which is the set limits are between 0.8 to 1 gram per decimeter cubed. If blood is not between these, if blood is too low, the hypothalamus will sense the blood glucose level, and also the glucose receptor cells in the pancreas will sense this. And if blood glucose level is too low, it's called hypoglycemia. If it's too high, it's called hyperglycemia. And anyways, there will be a negative feedback control loop that will happen. So let's introduce you to the different structures involved. We've got the liver and the muscle cells, where the glucose is stored as glycogen, and subsequently it can be broken down from glycogen to glucose when the body needs it. We've got the hypothalamus, which is the control center of, and it detects the blood glucose level. We've got the pancreas. Pancreas is subdivided into glucose receptor cells, or the endocrine cell. Now, the endocrine cells are cells that make up the islets of Langerhans, and these cells can be subdivided into alpha cells and beta cells. So alpha cells' main function is to produce glucagon, and beta cells is to produce insulin. Now, glucagon plays a role in glycogen to glucose, but also pyruvate to glucose. So glucose synthesis, basically, gluconeogenesis. Insulin, on the other hand, stimulates glucose uptake by a mechanism which I'll show you later, and it also stimulates conversion of glucose to glycogen. All right, so here, as you can see, here is the mechanism in which glucagon act on the cell. So this right here is the cell membrane of a liver or muscle cells. We've got glucagon as the primary messenger, and you've got the receptor on the cell membrane. Once glucagon binds to the receptor, there will be a conformational shape change in the receptor, which activates a G protein. This G protein will then move and activate another membrane-bound enzyme called adenylyl cyclase. And adenylyl cyclase synthesizes the conversion of ATP to cyclic AMP. Cyclic AMP is the secondary messenger. And once CAMP binds to inactive protein kinase A enzyme, there will be an enzyme cascade going on. As this protein kinase A enzyme is activated, it will phosphorylate a inactive phosphorylase kinase, which will activate it, and this active phosphorylase kinase will then activate the inactive glycogen phosphorylase, which will then, once become active, will catalyzes the conversion of glycogen to glucose, or glycogenolysis. Right, so that is basically how glucagon works. And this here is how insulin works. So basically the same thing. Insulin, this is also a liver cell. On the membrane, you've got insulin, and you've got in the receptor. Once the receptor, insulin binds to the receptor, there will be a secondary messenger sent out. And this secondary messenger will activate vesicles with GLUT4 channels. These vesicles will travel to the membrane and merge in with them, fuse in, which will introduce these channels for glucose to come inside the cell, right? Just like that. That's a net movement of glucose into the cell. Now we'll move on to the different diseases involved. So we've got, so these are the processes, you know, glycogenesis is when you convert glucose to glycogen. And glycogenolysis is the other way around, when you go from glycogen to glucose, right? So diabetes mellitus, this is the disease diabetes. And we've got two types, type 1 and type 2. Type 1 is an autoimmune disease where white blood cells attack your B cells of the pancreas. And because the B cells are involved in insulin production, our body won't have enough insulin, so glucose is not taken up, or not converted to glycogen to be stored. For type 2, it's not an autoimmune disease, but rather the result of an unhealthy, high energy, high sugar lifestyle, where you consume too much glucose, that your body produces so much insulin that the receptors on your muscle and liver cells become unresponsive to these hormones, the insulin hormones, and the insulin loses its effects, so the cells become resistant to insulin. So again, glucose cannot be uptaken by the cells, and we also end up with a lot of glucose in our bloodstream. And this is bad because there will be a lot of symptoms associated with this, and death can be even a consequence of diabetes. So first thing is high thirst, right? Because since there are more glucose molecules in your blood, the water potential in there will be lower. Therefore, water in cells will diffuse out into your bloodstream and increase the blood concentration of water, and your cells will lack water, so you'll always be constantly thirsty. You'll have poor vision because your eye pupils of your eyes have a lot of water, and again, since there's so much glucose in your bloodstream, water will diffuse out and a lot of structures will lose its healthy water potential. You'll urinate a lot because a lot of water in your bloodstream. Since glucose is not uptaken by cells, your body will have to break down other molecules like lipids and proteins to feed off your cells as energy. And you will also feel tired because there's not a lot, your cells are not uptaking enough glucose to be healthy. Right, so those are the symptoms associated. Now we'll move on to biosensors and test strips. So basically, these are how you would detect glucose blood level. Right, so test strips test for the glucose blood level in your urine, but the thing is, it only tests for glucose concentration higher than renal threshold. So in your kidneys, once blood passes through your glomerulus and the renal capsule, glucose will be ultrafiltrated. So the process is called ultrafiltration, which I'll cover in the next video. And once it's been filtrated out, it will a lot of important molecules like glucose, water, essential salts will be selectively reabsorbed back into the bloodstream. and the other waste molecules like used hormones or urea or other toxins or metabolic waste will be excreted out. But once glucose concentration reaches a level that it's too high, not all glucose will be reabsorbed back into your bloodstream. So a lot of glucose will end up in your urine, and you can use different ways to test the level of urine in your level of glucose in your urine. So test strips will only test for the concentration of glucose above the renal threshold, right? So that's when kidneys will stop selectively reabsorb the glucose back. So test strips have a pad at the end which have immobilized glucose oxidase and peroxidase enzymes immobilized onto it. Glucose will glucose oxidase catalyzes the reaction between glucose and oxygen to form gluconic acid and hydrogen peroxide. Hydrogen peroxide will then react with a colorless substance in the pad itself and will form a colored substance and water. This entire reaction is catalyzed by peroxidase, and the color ranges from teal to brown, right? So the darker the color, the more glucose concentration there is in the bloodstream. For biosensors, you'd also use glucose oxidase, but you're not using peroxidase. So what happens is there will be glucose oxidase immobilized onto the bottom. There'll be a partially permeable membrane at the top. So when you pour in blood or when your sample is blood, a lot of molecules will be filtered out. Only glucose will come through because it's small enough. And the glucose, once it comes into contact or binds to the glucose oxidase and react to form gluconic acid and hydrogen peroxide again. Hydrogen peroxide is then put through electrolysis, where it will be oxidized at the anode to release electrons at the cathode. And there'll be a computer amplifying the current, electric current. And there'll be also a computer reading it and converting it to however there are many grams of glucose per decimeter cube. So this is ways you can monitor your blood glucose level to keep it at a healthy level and not be toxic. So in the next video, we'll talk about blood urea concentration, and we'll talk about how the body measure these and how the body controls them.